This module, we're going to go over RNA-seq technology and the statistical methodology that's used to analyze it. I'm going to start with a very simple introduction describing how the technology works. So we have RNA transcripts. We convert them to cDNA. And once we do that, we can fragment it and sequence it. Once we sequence it, we have reads, just like with any other application. And once we have reads, we can, for example, map them to the genome. So just to give a very quick illustration, imagine you have a gene represented here with this box. And we want to know how much of this gene was present in our sample. And we actually have two samples, sample A and sample B. So each sample has a series of reads, and we can just simply find how many map by aligning each sample to this gene. So in this cartoon, we see that sample A has more gene expression for gene 1 than sample B. And you can do statistics on that, something we're going to cover later. So in a sense, we are back to the original problem that we described for microarrays. We have a big table, we have samples on the columns, and we have counts now for each gene on the rows. And if you have a table like that, then you can apply very similar approaches to the ones that we described earlier for microarray data. One difference is that now we're dealing with counts instead of just intensities. And for counts, most of the approaches that are currently used, we model with Poisson or negative binomial. Two examples of methods that do this are EdgeR and DESeq, two very widely used methods. Up to now, we have simplified the problem. It turns out that genes aren't just a piece of the genome. For humans, we have exons and we have introns. So the sequence of the RNA transcript aren't exactly represented on the genome. And in this cartoon, we see what happens. So here's the genome. In yellow, we see the exons. In black, we see the introns. And when RNA is transcribed, these introns are spliced out, and we create this RNA transcript that only contains the exons. Now, what does that mean? That means that we have these regions called junctions. Here's a close-up of those junctions. And the sequence of this junction doesn't appear on the genome. It has half of it, well, one part of it maps to one exon, for example, over here, and the other half maps to the other exon, for example, over here. So that is something that in this module, we're going to explain how we deal with that. A second complication is that the same gene can have many different transcripts. What does that mean? So a gene is something that is not completely well defined right now. But if you go to the databases where you're told what a gene is, you will find that it has a start and an end. But when you look a little closer, you will see that it'll include several versions of the gene where different exons are included or not. So in this example, we see that this particular gene has 13 possible transcripts, each one containing different exons. So one of the new possibilities that RNA-seq opens up is the ability to distinguish these transcripts. So you might have a sample where even though the gene expression for a given gene is the same in the two samples or the two populations, in one population you see an overrepresentation of one of the transcripts compared to the other. That's something that microarrays can't measure. But with RNA-seq, we can. So RNA-seq opens up the possibility of measuring these different transcripts. So while we have approaches like EdgeR and DESeq and DEXeq that basically count each gene or each exon, quantifies the amount, we also have techniques that try to determine from the reads what transcripts are present. Some of these actually try to do it from scratch without a reference genome. Example of these methods are Trinity, OASIS, Cufflinks, and Scripture. We're not going to cover the ideas behind these specific methods, but because they're somewhat more advanced than what we're doing in this course. But you should know that they're out there and they have software that you can try. So in the next modules, we're going to go beyond just counting reads per gene, and we're going to try to explain the ideas behind methods that try to quantify transcript levels for different transcripts in the same gene.